So it's an honor for me to introduce our next speaker on stage. His name is Nicolas Brunel, and he's a professor in theoretical neuroscience at Duke University and Bocconi University, and he's going to speak about the role of inhibition in cortical dynamics. Thank you for being here.
So first, I'd like to thank the organizers for putting together this fantastic meeting, and also a great choice for a venue. So I'll be telling you about a few stories about the roles of inhibition in stabilizing and shaping the response of cortical networks. And so we've known from, at least from the times of Santiago Ramón y Cajal, that neurons are very diverse and they come in different cell types. And a major subdivision is a subdivision between the excitatory neurons that we see here on the left, the cortical pyramidal cells, and inhibitory neurons. And of course, one of the major questions in neuroscience has been what are the respective roles of these different cell types. And in particular for inhibition, for a long time people have thought that inhibition could play a role in controlling the mean rate of excitatory neurons or maintaining excitatory-inhibitory balance, which would ensure that cortical operations have a low metabolic cost and also avoiding all kinds of instabilities that could lead, for instance, to epileptic states. And this question of excitation-inhibition balance came to the forefront of theoretical neuroscience research in the 90s, where people started to wonder why the cortical cells have this property of irregularity of their firing. So this is a very ubiquitous finding in cortex. And a number of groups proposed that this could be achieved in large randomly connected networks of excitatory inhibitory neurons, provided inhibition is strong enough. And so that led to this concept of balanced networks where each neuron receives a very large excitatory inputs and also very large inhibitory inputs. And it turns out that in these networks, these two automatically balance each other so that their mean inputs are actually can be below threshold, but then these networks also automatically generate large temporal fluctuations, which then lead to highly irregular firing. So balanced networks are in one extreme of possible models of cortex, like a very strongly coupled case where basically both recurrent excitation and inhibition are very strong. But of course, there are other models that have been proposed. Some models, for example, some models of visual cortex are essentially purely feed-forward, and they assume that that computations can be performed purely by feed-forward connectivity. And there are some intermediate regimes, of course, like the supralinear stabilized network by Ken Miller and collaborators. So what I'd like to tell you about a few stories that are all in collaborations with experimentalists, where we've used various types of manipulations of these networks, either optogenetic or pharmacological, to try to understand better this inhibition, stabilization, and how inhibition shapes the response of excitatory neurons. And so let me first go quickly through the first two questions that basically are published. So for people that are interested, they can just go to these papers. And then I'd like to spend more time on the last part, which is in preparation. All right. So it's been known for a long time also from basically an old paper by Misha Tsodix and collaborators that inhibition, there is a signature of inhibition stabilization is the so-called paradoxical response. So in inhibition-stabilized networks, it turns out that if you stimulate, so you provide an excitatory input to inhibitory neurons, that results paradoxically in the decrease of their rates. So one can see that in the simplest possible excitatory inhibitory network, a Wilson-Cowan type model, you can look at the phase plane. So this is the excitatory rates on the x-axis, inhibitory rates on the y-axis. These are the null clines of excitatory and inhibitory rates. And inhibition-stabilized networks have this characteristic cubic-shaped null clines. And for a network to be inhibition-stabilized, one needs to sit in this intermediate branch of the excitatory null cline. And then what happens is that if you freeze inhibition, in these cases, you see that this fixed point will be unstable, and you'll go either to a high rate state or a low rate state. Add in an excitatory input to inhibitory neurons shifts the nullcline, the inhibitory nullcline here in blue, up, and that leads to this paradoxical effect. Basically, this intersection goes down, so both excitatory and inhibitory rates go down. So a few years ago, in collaboration with Mark Heisted from NIH, we decided basically to test this, this prediction, and the idea is to understand, well, is mouse cortex an inhibition-stabilized network? And so the idea is quite simple. Basically, you need to have a mouse where all inhibitory cells express an opsin and then shine light on cortex, so you activate these inhibitory cells. So then what you expect are two possibilities, right? So if the network is not inhibition stabilized, you expect that the response of inhibitory neurons, will just go up monotonically with light intensity. And excitatory neurons will be suppressed by this increase in inhibition, so they basically at some point their rates should become very small. On the other hand, in an inhibition-stabilized network, you should rather see this V-shaped response of inhibitory neurons. Initially, you'll have this paradoxical suppression of their rates. At some point, though, the excitatory neural network will be fully suppressed, and so at this point, the network is no longer inhibition-stabilized because there is no longer any excitatory activity. So the inhibitory neurons then are just purely driven by light, so they should increase their rates. All right, so basically, so that's what Mark and his people from his group did, and in multiple cortical areas. I'll just be showing you what happens in V1, but very similar results were obtained in somatosensory and motor cortices. And basically, this is the result, the average response of excitatory and inhibitory units in these experiments, and you see that inhibitory units you do see this characteristic V-shaped response as a function of laser intensity. All right, so that's consistent with the fact that mouse cortex is an ISN. And note that these results are obtained at rest. There is no sensory stimulus. In fact, it's been debated whether perhaps cortical networks will become inhibition stabilized in the presence of sensory stimulation, but not at rest. But this really indicates that already at rest, they are inhibition stabilized. Another prediction that these inhibition-stabilized networks have is that transiently the inhibitory firing rate should go up before they relax to a rate that's lower than baseline. And we also verified that prediction in so the data is also consistent with that prediction. You have a quick, short latency increase in firing rates due to light. But then, basically, because the excitation is going down, that provides, there is a removal of excitation in inhibitory cells, and so they also go down. So it turns out that inhibition also profoundly shapes the response of networks in V1 to optogenetic stimulation of excitatory neurons now. And so this is a different set of experiments which were performed now both in Mark Heisted's lab, so again at NIH, but also in John Reynolds' lab at the Salk Institute. And the nice feature of this data is that we have very similar data in mice and monkeys, so I'll be showing you side by side. So what happens when you shine light on cortex, where now excitatory neurons express an opsin? So you would expect that basically excitatory neurons should go up because they are strongly activated by light. And that's what we see in some cells, but in fact, not all cells. So these are examples here in mice and monkeys. Here, the x-axis is the contrast of a visual stimulus. In some cases, in this experiment, they were also showing a drifting grading. But other cells are strongly suppressed, right? Indicating that basically they are probably strongly inhibited. So indirectly, light will indirectly activate inhibitory neurons and suppress the activity of some neurons. And there are also more complex neurons that are increased at low contrast, but decrease their rate at high contrast. All right, so these are just a few example neurons. Now, more interestingly, we see that in some cases, specific, and this is most pronounced at high contrast in monkeys, what we see is this very surprising finding that the whole distribution of rates across the networks is basically unaffected by this optogenetic stimulus, which is very surprising. First, we would expect an average an increase in average rates. We would also expect actually an increase in the width of the distribution, because after all, these inputs we can expect them to be uncorrelated with the visual stimulus. But in fact, that's not what we see. We see essentially this invariance of the distribution. So that's what we term the kind of reshuffling of firing rates. It's as if this additional stimulus maintains the distribution intact, but reshuffled rates across neurons. So how is that possible? Because we are in fact activate, or not we, but they are activated these networks with this strong excitatory input to excitatory neurons. Well, so we turn back to this kind of standard balanced network models. So we very simple randomly connected network of excitatory and inhibitory neurons. And in this network, we can vary the coupling strength and the way we can have all synapses be proportional to J. And so this is, we can vary J and then vary the strength of coupling. So we can look at what happens in a weakly coupled case. And by the way, this is the distribution of optogenetic inputs in these networks, and that mimics some experimental data showing that opsin expression could be well described by a log normal distribution. So what happens at weak coupling is what one would expect intuitively. So the distribution of rates increases, the mean increases strongly, and also the standard deviation of the rates increases strongly. This is the CDF of rates. And this is a scatter plot of like delta rate versus rate. You see a strong increase in excitatory firing rates. That's what you would expect intuitively, right? And the red points are excitatory, blue points inhibitory neurons. On the other hand, if you increase very strongly the coupling strength, so now we are in this very strongly coupled, essentially in the balanced regime, now what you see is very similar to what you see in the data. Now the light basically only very weakly affects the distribution of rates. And now the two fall on top of each other. And what you see here is that this scatter plot indicates that you have basically, so the mean is essentially unaffected, and you have an anti-correlation between the change in rate and the rate, which is in part due to this hard floor on the delta rate. Delta rate, of course, cannot be lower than minus the baseline rate. And so, in this network, we can also do some mean field theory calculations, and we can show that, in fact, in the strong coupling regime, the mean delta rate actually will go to 0 when k goes to infinity. So the number of connections per neurons go to infinity, and will go to 0 as 1 over square root of k. And we also understand through these calculations why the distribution is approximately invariant. All right, so that was kind of a very brief summary of these two first stories. So now let me move to the second one, which is the third one, which is the most recent. And now, so far, I've totally ignored the fact that inhibitory neurons are not a homogeneous population, and rather inhibitory neurons can be subdivided into multiple cell types. So this is a cartoon from a recent paper by the Allen Institute, where they extensively characterize all the possible synaptic connections in this kind of 4-population network. And of course, that leads to the next question, like what are the respective roles of these distinct inhibitory subtypes? And in particular, in the context of inhibition stabilization, one might wonder whether is there one particular cell type that's responsible for inhibition stabilization, and maybe others play different roles. And for instance, some people have thought that parvalbumin positive interneurons would be the ones that are responsible for stabilization. All right, so how to dissect the roles of distinct inhibitory cell types in cortex? So there are various methods that people have used over the years. One is cell type specific optogenetic, so express an opsin in a specific cell type, and then you can activate these cells or inhibit them, see what happens to the network. Another classic strategy is to use receptor-specific pharmacology, so you can manipulate receptors that are perhaps present in some cell types. So more recently, Mike Tadros, while he was at Janilia Farm, developed this strategy which he calls drugs actively restricted by tethering, or DART. The idea there is that you can express a particular protein, a halo tag protein, in a specific cell type. Okay, so that gives you the cell type specificity. And this protein, you have a flexible this flexible thing here that can bind to a specific drug. Okay, so then what happens is that when you inject this drug, so for instance, let's say an antagonist of AMPA receptors, the drug binds to this tether, and now it can block AMPA receptors, but only AMPA receptors that are present on that specific cell type. So now you have both cell and receptor specific both cell and receptor specificity. So that's what Lindsay Glickfeld, who's at Duke, used in experiments in mouse V1, and she chose to express the halo tag proteins in somatostatin cells and to use YM90K, which is an antagonist of AMPA receptors, onto somatostatin cells. So essentially what this drug is doing is weakening excitatory inputs onto somatostatin cells specifically. And so what you would expect then is that, well, somatostatin cells should become less active, they receive less excitation, and then as a result, pyramidal cells should be less inhibited, so they should go up. And that's what indeed you see in most cases. So now this is data, so this is now calcium imaging of somatostatin cells on the left and pyramidal cells on the right. And these are situations in which the animal is stationary, so it's not moving, and is basically exposed to a visual stimulus at various degrees of contrast. And this is, again, these moving gratings. And we see that basically here, looking at 25% contrast, what you would expect, right? So in control conditions, somatostatin cells are here in black, and when you apply DART, they reduced their activity, and vice versa, pyramidal cells increased their activity. Now, the first surprise is that this effect is strongly contrast dependent. So if you increase contrast, the somatostatin cells now are much less inhibited than in low contrast, which is already a first surprise. And then you would expect that then pyramidal cells are also less inhibited, right? Because the difference between the two gets smaller. Well, on the contrary, now they increase more their rates. So that's the first surprise. Now, even more surprising is what happens in running conditions. So now if you look at data when the mouse, because these mouse are on these, they can either be stationary or running in these conditions on a treadmill. And then what you see is that in running at high contrast, now the situation reverses. So somatostatin neurons are now more active in the presence of the drug. So it's another paradoxical effect, if you will, that you're reducing excitation onto an inhibitory neuron, and then the firing rate increases. And so these are summary plots in stationary and running conditions showing the amplitude of the calcium fluorescence as a function of contrast for somatostatin cells. So we've basically wondered how can this happen, and what are the mechanisms by which, in particular, we can see these changes in the response as a function of contrast and state. So we turn to the simplest possible model that could describe these results, which are basically a rate model, which now includes these 4 populations. So we have excitatory cells, then parvalbumin, somatostatin, and VIP positive interneurons. And we are using basically, there is potentially a 4x4 coupling matrix, but for simplicity, we are neglecting some of the weaker connections based on data by Massimo Scanziani, Allen Institute, and others. So this is not a full 4x4 matrix, but there are some missing entries here. So note that, of course, the crucial parameter here is WSE, the strength of the excitation to somatostatin cells. And this is the parameter that will be affected in the DART condition. We're just reducing WSE by some fraction, where X is the strength of DART. And now the question in the model, what happens when you do this? And importantly, how do the effects depend on all these parameters? Well, first, to basically quickly, we can reduce actually this network to a 2-population model, which simplifies a lot the analysis. And we have basically this W stars are parameters that take into account the effects of the other populations, parvalbumin and VIP. So for instance, WEE star would describe the feedback loop between pyramidal cells and parvalbumin positive interneurons. And you have WSS star takes into account the feedback loop with VIP cells, and so on and so forth. So let me go quickly. I don't have time to describe all these parameters. But at the end of the day, what we can do is basically, so first we realize that the effects only depend on 2 parameters of the network. So the strength of recurrent excitation, which is here the x-axis. Here the tilde, basically, tilde means that we are multiplying the W with the slope of the transfer function of excitatory cells. right? So this is the gain of the transfer function of the excitatory cells at the given rate that these cells have. And then on the y-axis, this is the strength of the inhibition of somatostatin interneurons onto excitatory neurons, and again, there is this tilde. Note that, so there are a few horizontal and vertical lines here. So this one is the line which defines the inhibition stabilization. So on the left, the network is not inhibition stabilized, right? There is this very simple condition in these networks, and on the right, it's inhibition stabilized. There is this other vertical line here, which is the line, so on the left, these networks are inhibition stabilized even in the absence of somatostatin cells. So only they stabilize purely by PV cells on the left of this line. On the right, on the other hand, PV cells are not enough to stabilize the network. You also need these other type of cells, the somatostatin positive. And I don't have time to tell you about this region down below, but in any case, we don't see this behavior is not compatible with what we see in the data. All right, so what do these two regions produce in terms of what happens during DART? So these are these small cartoons show the null lines now of in the RE-RS planes, and the black line is the somatostatin null line. Okay, so somatostatin neurons increase their firing rates as a function of the excitatory rate. However, in DART condition, of course, this relationship becomes weaker, and so the slope of these curves goes down. The green curve now is the other null line, the excitatory null line. And it turns out that, I mean, this is very simple to see that, but in this region, the excitatory null line has a negative slope, while in this region, it has a positive slope. And so that changes now what happens in the presence of DART. So region 1 is what you would intuitively expect. Both, so somatostatin goes down, excitatory neurons go up, while in this region, actually, both types of neurons go up when you do this manipulation. And what's important to note here is that you can actually move in this plane both by changing visual input and or state, because changing the inputs to this network actually change the gains of these populations, the phi primes, because you're changing the inputs, and so you're changing the gains when you have nonlinear transfer functions. And so that allows you, in fact, to move from one region to another as we're changing either a contrast and or state. And in fact, we showed that the data in the stationary state are consistent with the network being in region 1 here, but not far from this vertical line, while in running conditions, then the network goes actually in the region where somatostatin interneurons are also required for stability. And in fact, we can fit the data quantitatively very well using this model. And that's basically the fit indicates, if we make a zoom on this little corner here, these are the 3 contrasts in stationary conditions, and these are the 3 contrasts in running conditions. So basically, at high contrast and in running, we cross into this region where somatostatin are also needed for stabilization. All right, so let me conclude. Basically, experimentally, we see that weakening AMPA receptors onto SST cells is actually sensory input and state dependent. And that leads, in fact, to a new paradoxical effect where weakening excitatory inputs onto a specific cell type actually leads to increase of its activity. And we've shown in this model that this is actually equivalent to the network being stabilized by SST neurons. So SST neurons are actually needed for stabilization in this case. And in fact, we think that this tool will be very useful in order to dissect also the roles of other inhibitory cell types. And so hopefully we'll get more data to further constrain the regime in which cortical neurons are operating. In fact, and also how this changes as a function of sensory input and state. All right, so let me conclude. So I'd like to thank all the people that have worked on these different projects. The projects involving optogenetics and inhibition stabilization and reshuffling. There was a collaboration with Alessandro Sanzeni, who's now actually at Bocconi University, and we've also collaborated with a group at Columbia, Agostina Palmigiano, who was a postdoc with Ken Miller. She's actually now just moved to UCL, and Tuan Nguyen is a PhD student of Ken. And we we were lucky to benefit from this collaboration from experimentalists Mark Heisted for mouse data and John Reynolds for the monkey data. And the last part about the cell type specific pharmacology, this is a Duke collaboration with, so on the theory side was spearheaded by Yingming Pei and David Saint-Amand, and all the experiments were done by Céline Camarata in Lindsay's lab, and we were using these DART compounds that were provided by Mike Tadros. Well, thanks for your attention.
Thank you for your talk. I was wondering in the first part where you talk about ISNs and V1, if in addition to subdividing the cells into cell types, if you've looked at how tuning affects it, so co-tuning of inhibitory neurons and excitatory neurons, or like opposite tuning, since that is also a feature of cortex.
Yeah, so unfortunately these experiments were in the absence of any sensory input, so there were no information about the tuning of cells, but it will be interesting to see, in fact, what happens as a function of their tuning.
And did you also, I noticed you only looked at the sensory areas, but you also have the association areas. Did you look at if there's a difference in like retrospinal cortex or parietal cortex?
So basically, this slide shows all the areas. So basically, there are only 3 different areas that were explored by Mark and his collaborators, so V1, somatosensory, and motor. The 3 gives very similar qualitatively similar results, though the strength, I mean, the magnitude of this dip varies a little bit between these regions and is less pronounced in motor cortex for some reason. But apart from that, it's very similar.
One last question.
Yeah, thanks for this nice presentation. This data from this Campagnola et al. Paper from Al Institute that you cited, now, they also show that each of these connection types have a characteristic short-term depression or short-term facilitation.
Yeah.
Do you think this has an impact on this phenomena that you studied?
Yes, so right, and of course, so this is one of the scenarios we considered to explain this data, that potentially maybe you could move because of short-term depression and or facilitation. That could be also another way, in fact, to move on this plane, because effectively now this would not be through the nonlinearity of the transfer function, but basically effectively you would change the weights themselves. So we chose to use we basically chose this strategy, but we also considering looking at what happens in the presence of short-term plasticity, and it's certainly another way in which you can move in this plane. But we haven't really yet checked whether that can give rise, especially if the Campagnola data is consistent with the type of changes on that plane that we see in the data. But yeah, that's a very good question.
Okay, so we need to finish up here. So let's thank Nicolas again.